Hello, my name is Muhammad Ali. I am pharmacist by profession. I have nearly 40 years of experience working for pharmaceutical industry. I worked with big names like Johnson and Johnson and Novartis. My work was mostly on floor. I also worked in laboratory too. During my work on production floor, I realized that cleaning of equipments is very vital process in the manufacturing of medicine. Thorough cleaning is very important. If cleaning is not done properly, there is high risk of inter-batch, inter-product mix-up. This initiative of mine will also help the fresh pharmacy graduates who like to make their career in pharmaceutical industry. In this training modules, I will cover the different requirement of cleaning validation, individual steps for validation of cleaning procedures. Here I will also give some hints and tips related to the cleaning validation process. This training module is intended for people in production, QA and QC participating in validation of cleaning procedure. Now, the cleaning of equipment is the requirement of FDA. FDA expects that firms to have written cleaning procedure for all the equipments which comes in contact with the product. FDA expects firms to have written cleaning validation master plan for all the equipments. FDA expects firms to have written validation protocol, and also the cleaning procedure must have and also they expect that cleaning procedure must have been validated and documented. All their cleaning procedure must have been validated and documented. This is the FDA requirement. Let's first understand what is the validation and what is expected from the validation. Let's suppose we have a cleaning procedure with us wherein we are using water, disinfectant, we are also scrubbing to wipe off the hard material, we are also applying disinfectant to disinfect our equipment, we are also applying 70% alcohol, isopropyl alcohol to disinfect our equipment. So we have the cleaning procedure with us now. Now we want to know if this cleaning procedure really working. Is it really cleaning the equipment? Is it really removing the previous product traces? Is it really disinfecting the equipment to such extent as we want? In order to confirm that our cleaning procedure is cleaning the equipment as needed, we have to validate our process. We have to validate this cleaning processes. So the definition of validation is establishing the documented evidence that a process does what is claimed to do. What is expected from the cleaning validation? A validation process has to provide scientifically justifiable data that shows that the system consistently performs as expected and produce a result that consistently meet the predetermined specifications. Before we start how to validate cleaning procedure, let us discuss the situation where there is low risk exists and where there is high risk. In case of automated cleaning procedure, the risk is very low. In case of manual cleaning, the risk is very high. Similarly, in case of simple equipment, the risk is very low. In case of complex equipment, the risk is very high. Now, for less potent product, the risk is very low, and if we are processing highly potent product, the risk is very high. Low risk production products are non-sterile, low potency products, while high risk drugs are sterile and highly potent products. It also depend on this also depend on the type of the facility. If we have dedicated facilities, risk is very low. If we have multi-purpose facilities, risk is very high. Risk also triggers because of the production type. If we have campaign production, the
the risk is very low. If we have batch-wise production, the risk is very high. This is a summary of the risk. In case of low risk, automated cleaning, simple equipment, low potent product, dedicated facilities, combined production, large batch size. As far as high risk is concerned, it's because of the manual cleaning, complex equipment, highly potent products, multi-purpose facilities, batch-wise production, small batch size. Before we start cleaning validation, we should address following questions. We should do some groundwork before we start cleaning validation. We must know when cleaning validation is needed. We should know what items are being removed during the cleaning of the equipment. From where to draw the samples. We should know the sampling spots. We should know what kind of analytical test is needed to verify the equipment is clean as per our requirement. What kind of test procedures to be adopted. We should also know what kind of physical parameters shall be evaluated. We should also know how much residue from the previous product is allowed in the subsequent product.
Now let us talk about when cleaning validation is to be conducted, when it should be performed, when it is really needed. Whenever you have a new cleaning procedure, you need to do the cleaning validation. Whenever you have new product, you need to do the cleaning validation. Similarly, when you have a new equipment, it requires a new cleaning procedure, especially designed for this new equipment. And here you also need to do the cleaning validation. In case of major change in the equipment, you should do the cleaning validation. Likewise, when you change the cleaning procedure, like if you want if you will change detergent or detergent quality or brand of the detergent, you need to do the cleaning validation. Let's discuss what are being removed during the cleaning procedure. During cleaning procedure, active ingredients are removed. During cleaning procedure, excipients are being removed. Detergents is being removed. Microbes are being removed. In validation of cleaning procedure, we have to test these to confirm that our cleaning procedure does what it is expected from it. In other words, we are validating our cleaning procedure. To validate our cleaning procedure, we need to test the surface of the equipment for the traces of the active ingredients of the previous product. For this testing, we need to draw samples. For sampling, the following techniques are being used. Swab. Samples are taken by swab to test the traces of the active ingredient, to test microbiological contamination. Sometimes we have to test the final rinsing of the water for the traces of the previous product. Sometimes, if the active is not soluble in water, we have to extract it by solvent for its testing. Hence, these techniques are used for drawing of the samples: swab, final rinsing, and solvent. Now, to test the traces of the previous product active, we have to use an analytical procedure which is very sensitive, so that we can test the lowest quantity of the material of the previous batch. So in case of analytical procedure, its limit of quantitation should be less than allowable carryover. I will talk about the allowable carryover in my next slide. Hence, the most important question is how much residue will be permitted? How much residue of active substance of the previous product is allowed in the subsequent product being produced in the same equipment? This is the most important question for the validation of the cleaning procedure. So in my next slide, I will talk about this
how to calculate the allowable contaminant. Before we start the cleaning validation, we must select the most difficult to clean product among the products which we are manufacturing in the equipment. There are many ways to find out which of our product is most difficult to clean. It is obvious that if active of a product is soluble in water, it is very easy to clean it from the surface of the equipment. But if the active is not soluble in water, we have to apply more physical pressure, more detergent, etc. to clean it. In this case, the cleaning is difficult. So solubility of active in water matter a lot. If it is not soluble in water, it is most difficult to clean. Sometimes the operator experience is also considered. Here he and she will tell you which product is most difficult to clean. There are many ways to do scoring and to find out difficult to clean product. Product which gets more score is considered most difficult to clean product. Secondly, the ratio of active to mass of the tablet is also used for scoring. Let's say that if we have a product where active per tablet is 10 milligram and the entire weight of tablets is 100 milligram. In other case, if we have 50 milligram of active in 100 milligram of tablet, we can say that the product with high strength is most difficult to clean. So there are different ways to score to find out which of a product is most difficult to clean. We have to select most difficult to clean product for our cleaning validation studies. I would like to stay more on this slide so that you can understand how to do scoring to find out which is most difficult to clean. If the concentration is less than 10 milligram per gram, the score is 1. It is easy to clean in this case. But if concentration is 500 milligram per gram, the score will be 4 and it is most difficult to clean. Similarly, if it is very soluble in water, the score will be 1 and it is not so difficult to clean as compared to practically insoluble.
Now let's talk about our main concern, that is how to calculate to what extent and contaminants from the previous product is acceptable in the next product. We have to find out how much quantity of the previous active is allowed in the next batch produced in the same equipment. So this is very important calculation and we are going to do that. Now, how to calculate acceptance criteria? Companies must demonstrate that the cleaning procedure routinely used for cleaning of equipment must be rigid enough to remove the traces of active of previous product to such extent that it is less than the acceptable limit in the next product. Contaminants must be less than the acceptable limit in the next or subsequent product. The limit established must be calculated based on sound scientific rationales. In the next slide, I will provide practical guidance as how to calculate the acceptance criteria. Method for calculating acceptance criteria. The following method is based on therapeutic daily dose. The principle for the requirement is that the standard therapeutic daily dose of the next or subsequent product may be contaminated by no more than a certain portion or proportion of the active of the previous product. Allowable contaminants from the previous product in the next product is less than 1,000 part of the active of the previous batch. As I mentioned earlier, that allowable contaminants from the previous active must be less than 1,000 fraction in the next or subsequent product. This 1,000 proportion is a safety factor. Here is the justification of the safety factor. For the first decimal, the drug preparation are generally considered as ineffective if the dose is only 1/10 of the dose. Second decimal, pharmacological safety factor. Third decimal guarantees a rugged validation program. Now, how to calculate the acceptance criteria? Let's suppose that we are producing three different type of the products, product A, B and C. The strength of product A is 10 milligram per tablet. Strength for product B is 20 milligram per tablet, and strength for product C is 25 milligram per tablet. Now the mass of one dose or the weight of the tablet for product A is 100 milligram. For product B it is 200 milligram. For product C it is 250 milligram. Maximum daily dose for product A is 1 tablet per day. For product B it is 2 tablets per day, and for product C it is 2 tablets per day. Hence, mass of maximum daily dose will be for product A 100 into 1 is 100 milligram, since we are taking only 1 tablet per day, and for product B it will be 200 milligram into 2 is 400 milligram because we are taking 2 tablets per day, and for product C it will be 250 into 2 500 milligram as we are taking 2 tablets per day. Now as far as batch size is concerned, for product A batch size is 80 kg, and for product B it is 60 kg, and for product C it is 50 kg. So minimum batch size is 50 kg. And equipment surface where we are mixing all these three types of the product is surface area is 200,000 square centimeter. So key criteria for limit calculation is 10 milligram because this is the minimum therapeutic dose, and maximum daily dose or mass of the maximum daily dose is 500 milligram, and batch size, minimum batch size is 50 kg, and the surface area of the equipment is 200,000 centimeters square. Now we have the data with us. The summary of data is as follows. Single minimum therapeutic dose of previous product is 10 milligram. Minimum batch size of the subsequent product is 50 kg. Maximum mass or daily dose of the subsequent product is 500 milligram. Maximum surface area of the equipment is 200,000 square centimeter, and the sampling surface is 100 centimeters square. Now here is the formula for calculation of maximum allowable carryover or contaminants. Single therapeutic dose of previous product is multiplied by minimum batch size of the next product, and it is divided by safety factor, and the whole thing is divided by therapeutic daily dose of the next product, mass of the daily dose of the next product. Now I have put all the data into the formula, and here's a single therapeutic dose of previous product is 10 milligram. I applied the safety factor, that is 1,000 of it, and the largest daily dose of the subsequent product, that the mass of this subsequent product is 500 milligram. Batch size of the subsequent product, the lowest batch size is 50 kg. Since everything is in milligrams, so I have to convert 50 kg into milligram. So the batch size will come up to be 50 million milligrams. So I put all the data into my formula, and the maximum allowable contaminant is 1000 milligram or 1 gram, or you can say 1 gram. So 1 gram of the previous product is allowed on entire surface of my equipment, which is which has the surface area of 200,000 square centimeter. Now for sampling, I have to draw sample of 100 centimeter square from the surface of the equipment. So sampling limit will be 500 microgram. Now let's talk about this is about the equipment and the sampling site. And this is the diagram for the collet crawl. And here I have mentioned where to take samples for chemical tests and where to take samples for the microbiological tests. We have to take samples from the inner surface of the equipment for chemical tests as well as for microbiological tests. We have to take samples from the mixing paddle, which is difficult to clean, and we have to take samples from the places which are very difficult to clean for chemical tests as well as for microbiological tests. Here I also mentioned the hot spot and critical site. But these two terms I will describe in the next slide. Now, hot spot is a spot if not cleaned properly will contaminate entire batch. Example, arm of the mixing paddle. If arm is not cleaned properly, it will contaminate entire batch. As far as critical site is concerned, is site if not cleaned properly will contaminate a portion of the batch, not the entire batch. Example is the outlet of the mixer. So let's go back to our previous slide. Now here you can see that hot spot is the spot on the mixing paddle. If it is not properly cleaned, it will contaminate entire batch. As far as critical site is concerned, it is near this outlet of the equipment. If it is not properly cleaned, it will contaminate only a portion of the batch, but not the entire batch, as compared to the hot spot where it will contaminate the entire batch. So I have already explained you what is meant by hot spot and what is meant by critical site. Now here is how to take samples for the chemical test, and as far as microbiological tests, we have to use coupon for that test. And as I told you before, that we have to apply a very sensitive method for analysis of traces of the active ingredient. In the end, I would like to quote Galileo, which says, "Measure the measurable and bring immeasurable to measurable." Thank you very much.